Okay. Karibuni tena watazamaji wote wa Dr. Mlelwa Lectures kutoka popote pale ulipo. Unapofuatilia Dr. Mlelwa Lectures na wenda umekuwa ni subscriber, umekuwa ni follower wa hii channel, umependa kazi zetu, umependa vile ambavyo tunafanya, kwa hiyo umetaka na wewe mmoja kati ya mtu ambao unafuatilia kazi zetu. Actually we are very interested kwamba uko pamoja nasi na tuko tunaendelea na kazi hizi. Unaweza kaona kwamba hapa tuko tuna ni maandalizi ya notes za homeostasis and excretion. Kwa hiyo mimi niseme tu wote ambao mnafuatia Dr. Mlelwa Lectures, most of time tunavyokuwa silent you think as if tuko silent, but no, tuko silent by that meaning tuko tunaendelea prepare very good things for you. You can see hapa notes za advanced level homeostasis, notes nzuri sana I'm sure kwamba hujawahi kusoma kwa notes kama hizi. Notes nzuri sana, the diagrams are clear, the arrangement of concepts and different things, different concepts have been explained. Kwa mfano naweza nikasema sijajua sana sehemu ambayo inasumbua sana kwenye homeostasis and excretion hapa ni ipi, lakini let I move, let's say kuna baadhi ya parts ambazo nimechukua, nimemix some books for your advantage, kwa sababu vitabu mbalimbali vinakuwa vimeandika in different ways. Kwa hiyo manake nikimix different books inakuwa ni easier for you to understand rather than using only one book. So let's say nieleze nini hapa, nieleze kitu gani hapa so that you can get interested. Tafuta kitu kimoja hapa nieleze ili kwamba upate kuona I've used different books, so that's very interesting. That's very interesting to me because it will enable you to answer different questions at the same time. Let's explain the ornithine cycle, the mechanism of urea formation. I know that some of you know it in a different way, and even myself once upon a time nilikuwa nimefundisha kwa different notes. Hii mechanism nimeikuta kwenye kitabu cha Tai, and I've been interested with this mechanism because it explains the real process how it happens, because the formation of ammonia doesn't only happen in the cytoplasm, but it also happen in the mitochondria. It starts in the mitochondria and then it continues in the cytoplasm of the hepatocytes, which are the liver cells. So we call this the ornithine cycle. The process of urea formation occurring in the urea cycle, which is also called the ornithine cycle. Ornithine is one among the compound which is involved in this reaction. So the formation is somehow long as compared to that which is written in BS. And remember, there are some parts about BS or understanding biology, they have just summarized it, but actually zile parts ziko kwa undani zaidi wa pale. Now regarding the formation of urea, the process starts in the mitochondria, where the carbon dioxide from respiration combined with the ammonia. We call this stage the formation of the carbamyl phosphate. It starts in the mitochondria. So before the cycle, ammonia from metabolism of nitrogen containing compounds combined with carbon dioxide gas from respiration in a solution form. So it forms, that means the ammonia, it exists in solution form, which is ammonium ion NH4 positive ammonium ion, ammonia in solution form with C ammonium ion, and carbon dioxide in solution form it will be the hydrogen carbonate ion. As we know, carbon dioxide can combine with water to form weak carbonic acid, which can dissociate to form hydrogen carbonate ion. So we expect this ammonia in aqua solution, which is ammonium ion and hydrogen carbonate or bicarbonate ion, to combine and it results into the formation of the carbamyl phosphate. And this reaction is catalyzed by the enzyme carbamyl phosphate synthase 1. The reaction occurs in the mitochondria of the liver cells and requires 2 ATP molecules. So this mechanism or this stage of the Ornstein cycle is not written in the BS or understanding biology. You need to know that authors of different books, they tend to correct materials from different sources, including Google and elsewhere, in order to make sure that they get suitable materials for the better performance of a student. So after the synthesis of the carbamyl phosphate in the mitochondria using 2 ATP, what happens the next stage is the synthesis of citrulline. The carbamyl phosphate formed in the first step entered the Ornstein cycle and combined with the ornithine resulting in the synthesis of citrulline. So carbamyl phosphate, it entered the ornithine cycle by combining with the citrulline, aided by the enzyme citrulline synthase or ornithine transcarbamylase. In the reaction, the phosphate group is released, and the citrulline can easily pass through the mitochondrial membrane. Thus, it diffuses from the mitochondrion into cytosol. So these two stages, the formation of the carbamyl phosphate and the synthesis of citrulline, they occur in the mitochondrion, and then after that, the citrulline diffuses into the cytoplasm of the liver cells. We call them the hepatocytes. From there, the next stage is the synthesis of the agenosuccinate. So in the cytosol, in the cytoplasm, the citrulline combines with the amino group of aspartate, or we call it as aspartic acid. Aspartate is just a normal amino acid which we studied it in biochemistry. So in the cytoplasm, citrulline combines with the amino group of aspartate under condensation reaction catalyzed by the enzyme agenosuccinate synthase. This condensation reaction, so it involves the removal of water. And to form agenosuccinate, it requires ATP, which is hydrolyzed to adenine monophosphate, resulting in utilization of 2 high energy bonds. That means here it's not 30.68 kilojoule per mole, it is 61.2 kilojoule per mole they are released. Hiyo tunaisoma vizuri sana katika topic ya respiration, namna ambavyo hydrolysis of ATP na release energy, and if ATP is hydrolyzed in one bond, how much energy is released, and if 2 bonds they are hydrolyzed, how much energy is released. Magnesium ion acts the cofactor of this enzyme agenosuccinate synthase. This reaction incorporates the second nitrogen from aspartate. That means in the normal ornithine cycle mentioned in the biological science book, it seems as if both 2 amino groups or both 2 ammonia, they are coming from deamination, but it's not true. One of the amino group, it comes from ammonia from deamination or metabolism of nitrogen containing compounds, but one of the amino group, it comes from the aspartate or aspartic acid, you see. So it requires ATP, and this reaction incorporates the second nitrogen from aspartate. The fourth reaction, or the fourth stage, is the cleavage of the agenosuccinate, which was formed in the third stage. Now the cleavage of the agenosuccinate involves the enzyme agenosuccinase, an intermediate enzyme in the urea synthesis pathway whose function is imperative to the continuation of the cycle. Now it acts reversibly to cleave agenosuccinate into free arginine and fumarase. Remember, fumarase is one among the intermediate of the Krebs cycle. So arginine will continue with the ornithine cycle, but the fumarase can enter the Krebs cycle, or it can be converted to other carbohydrate compounds. The arginine continues with the cycle in the other stage, where the fumarase entered the tricarboxylic acid cycle, or TCA, or Krebs cycle. Remember that different metabolic activities in the body, they tend to interact. So there is no any metabolic activities which occur in its own. Metabolic activity, they tend to interact. You can see here the fumarase compound, which is formed in the ornithine cycle, it entered the Krebs cycle. That means there is interaction between the Krebs cycle and the ornithine cycle. From there, the cleavage of arginine, which was formed in the fourth stage. Arginine is hydrolyzed into ornithine and urea. Hydrolysis reaction involves the addition of water under the influence of the enzyme arginase. Hence, arginine is also known as the semi-essential amino acid. Though it is synthesized in the body, it is not available for protein synthesis. Ornithine is regenerated in this step, and the urea cycle completes by the formation of urea. The ornithine produced is transported back to the mitochondria, because the cycle starts in the mitochondria. To start the cycle again, while urea is transported to the kidney through blood vessels to be excreted. Thus, the urea cycle brings two amino groups. One from the deamination, or from the breakdown of the nitrogen containing compound, and one is from the amino acid aspartate in the third stage. It brings these two amino groups and the bicarbonate group together to form urea. Now, this is the detailed ornithine cycle occurs in the mitochondria and also in the cytoplasm of the liver cell. In the mitochondria, carbon dioxide combined with the ammonia from deamination under the enzyme carbamoyl phosphate synthase 1. Then we have N-acetyl glutamate, which is allosteric activator. I think for those who studied well biochemistry, they know what is allosteric. Activator. Now, under the enzyme carbamoyl phosphate synthase 1, carbon dioxide and this ammonia combine in the presence of 2 ATP to form carbamoyl phosphate, which enters the cycle. It combines with the ornithine and the enzyme ornithine transcarbamylase to form citrulline, and this citrulline into the cytoplasm. It combines with the amino group from aspartate. Then this forms it uses ATP, and then we get agenosuccinate. Agenosuccinate tend to combine with agenosuccinate is broken down into the arginine and the fumarase, under the enzyme agenosuccinase. Then arginase, it hydrolyzes into ornithine and urea. It hydrolyzes arginine into ornithine and urea. This urea travels to the kidney for the excretion, and then the ornithine enters again the mitochondria. This boundary, remember here, it notes the mitochondria. This boundary notes the mitochondria. So this molecule leaves the mitochondria, and this molecule enters the mitochondria. We have a lot of good stuffs, well explained diagrams, a lot of detailed mechanisms. We have a lot of them here, and I'm here also to show you that we collect these notes in different ways. Don't just think that it's easier process. We are using different resources, including Google. We are just downloading these diagrams, then going to the process of editing them, making them looking well, editing them in different ways, and then after editing them, you can see them these images, they have different modifications done. You can see, for example, this image, it has some writings here. We tend to raw it. Okay, I know not everyone of you are interested, but I just want to show you the process, how we are doing to make the book looking well. And I am a biology expert, but also an expert in graphic designing. If you don't want to learn biology from me, you can learn graphic designing. You see, things are looking like this way. So the appearance of the picture actually changes. We can magnify and doing a lot of things regarding this image. We can do things like hue and saturation. We can change the color of the image and different things in order to make it in a color that we are interested. So All in all, what I can say that we are doing different processes. We are doing different processes to edit these images. Very, very, very different processes. You can see we are correcting materials from website like this. Britannica, very famous website, I tend to use it. You can see these images also there from that website. Some questions from Koala, get them with their answers the way the kidney it is. This is the kidney from postmortem, kidney MIT up, and this is the little kidney of human being. You can see we are collecting actually a lot. This was from the homeostasis topic. A lot. Actually, what I can say that get tuned, stay tuned. We have a lot of things which we prepare for you. You can see I collected the notes for homeostasis. So I am collecting materials for you, and I'm sure that this book will help you to study easily and to answer well your exams. So thank you, and let me wish you nice studies.